Chapter 9 is all about genetics, or the inheritance, or the laws of inheritance. So how are traits passed down from one generation to the next? And the ancient Greeks had some ideas about this, but because they didn't have the scientific method, they never really did any experiments to find out if they were right or not. And our understanding of this starts with Gregor Mendel around 1860. So what Mendel did was he took pea plants, and he had pea plants that were true breeding. So this meant that they passed down the same traits from one generation to the next, and they never, the offspring always looked the same as the parents. And these pea plants are hermaphrodites. This means they have both the male and female organs in the same individual. And we'll see why that's important in a minute. So what Mendel did is he took these true breeding plants and he crossed them. So he was able to do that because he was able to take the flowers of the plants, and he was able, before the pollen became mature, he was able to cut off with the scissors the male part, and from another plant allowed the pollen to mature, and then brush the pollen onto the female part. And in this way he could cross different true breeding plants. And so he did this for a number of different traits, but we're just going to look at one of them, and that's the flower color. So he took true breeding plants that had purple flowers, and he crossed those with plants that were true breeding for white flowers. And you might think, well, if he crossed purple with white, maybe the offspring will look like a blend of the two. Maybe it'll be a lighter purple. But when he did this, he found that all of the plants, all of the offspring from this cross, looked exactly the same as the purple parent. They weren't lighter, they were exactly the same darkness of purple. And then the next thing he did was he let these purple offspring, he let them self-fertilize. So normally with this type of pea plant, the flower is enclosed and starts to self-fertilize. That is, the pollen from the male part lands on the female part, fertilizing the eggs in there. And that can all happen within an enclosed flower before it even opens. And so that's called self-fertilization. And he allowed that to occur. And then he observed how did the offspring look. And what he found is that although the white flower trait disappeared in the F1, in the offspring from the parents, in the F2, in the next generation, it reappeared again. And he got a 3-to-1 ratio of the purple flower to the white flower offspring. So what is the explanation for this? How could you get this characteristic, the white flower disappearing in one generation and appear in the next generation? The explanation is that there's actually 2 copies of each gene. So here we're showing the sperm cells from the purple flowered true breeding parent and the egg cells from the white flower parent. So the sperm have both, all the sperm have the large P because because the true breeding parent has 2 copies of the large P allele. So remember from the last chapter, you can have different versions of the same gene, 2 different alleles on the homologous chromosomes. So imagine these are the homologous chromosomes. They can either be different or the same. In this case, they're the same because it's a true breeding plant. it's the two homologous chromosomes have the same large P allele on the two homologous chromosomes. And so when this undergoes meiosis, all the sperm have big P. So I've drawn that here. And on this side, we've got the two possible eggs. And they're both little p, because it's the true breeding white flower parent. And so what you do is you do what's called a Punnett square. So the Punnett square shows you all the possible ways of combining the eggs and sperm. So in this case, in the case of the parent, in the case of the true breeding parent, it's very simple because there's only one possible type of sperm and one possible type of egg. And so what you do is you draw it out and then you show the combination of these two, sperm and egg. So here we're getting big P from the sperm and little p from the egg. And in this square we're getting also big P from the sperm and little p from the egg. And then in this square we're getting big P from the sperm and little p from the egg. And again, we're getting big P from the sperm, little p from the egg. So all of the offspring have the same inheritance in this case. Big P, little p. Big P from the sperm, little p from the egg. So all the offspring have one big P allele and one little p allele. We call the different alleles present the genotype, and we call the appearance of it the phenotype. So why is it purple? It's purple because the big P allele masks the little p allele. We say that the big P allele is dominant to the little p allele. Now in the F2 generation, now we're taking these big P, little p, F1 plants, we're self-fertilizing them. So basically it's this cross. Big P, little p times big P, little p. So you're really crossing the same genotype. This cross is called a monohybrid cross because each parent has one big P and one little p. We call that the heterozygous genotype. And so this monohybrid cross, when you let that go, here's the Punnett square. So in the sperm of the male parent, you've got big P, little p. And the same in the eggs, big P and little p. And so when you fill in your Punnett square, here you've got big P from the sperm, big P from the egg. In this square you've got little p coming from the sperm and big P coming from the egg. And in this square you've got big P coming from the sperm, little p from the egg. And in this last square you've got little p from the sperm and little p from the egg. So now you can see that there's really three different genotypes there. There's two big P's, there's big P, little p, and there's two ways to get that. And then there's two little p's. But there's really only two phenotypes, because as long as you have at least one big P, then that's purple flower. So big P, little p is purple, as is big P, big P. But little p, because it doesn't have any large P allele, that's white. So you got three squares with at least one big P, you have one square with no big P's, and so you've got 3:1 ratio of purple to white. And so from the results of this monohybrid cross, Mendel arrived at his first law, and that's called the law of segregation. And it says that there are two copies of each gene and they separate or segregate during meiosis. So when the sperm and eggs form, there's only going to be one copy of these in the offspring. So we have the parent, and then when that undergoes meiosis, there's only one copy in each gamete. And then in fertilization of the egg by the sperm, the two are combined again. That's Mendel's first law. Now we move on to Mendel's second law, the law of independent assortment. So Mendel's second law, the law of independent assortment, says that the inheritance of one character has no effect on the inheritance of another character. Or put another way, each of the alleles separates independently of other pairs of alleles during gamete formation. So remember in the last chapter we saw the mechanism behind that was the independent orientation of the chromosomes on the metaphase plate. But Mendel didn't know about the metaphase plate or about chromosomes because he was doing his work in the mid-19th century, and chromosomes weren't discovered until the early 20th century. So what was the evidence for this? What was the evidence that Mendel found? Well, it was called the dihybrid cross. So Mendel figured this all out through the mathematics of the crosses that he did. And in this case, in the case of his second law, it's the dihybrid cross. So this is shown here. In the dihybrid cross, you've got two genes and they're both heterozygous. So you've got a doubly heterozygous parent and another doubly heterozygous parent, and you're crossing them. So here is shown the Punnett square for that. So in a doubly heterozygous parent, there's 4 possible gametes. So in the monohybrid cross, we saw that when you're heterozygous for one gene, then there's 2 possible gametes, right? But if you're heterozygous for 2 genes, now there's 4 possible gametes, and those are shown here. So for the male parent in the pollen, you've got a sperm cell with big R, big Y, and you've got one that has little r, big Y, and a third that has big R, little y, and a fourth has little r, little y. Now these 2 genes in this example are called R and Y genes. The R gene controls the shape of the seed. So the big R allele means a round seed, and the little r allele means a wrinkled seed. And the big R allele is dominant to the little r allele. The other gene is the Y gene, and that controls the seed color. So the big Y allele is yellow colored seed, and the little y allele is green seed. So the big Y allele is dominant to the little y. So if you have a heterozygous for the Y gene, that's going to be yellow seed. If you're heterozygous for the R gene, it's going to be round seed and not wrinkled. So here we have the Punnett square. Now this Punnett square has 16 squares because there's 4 possible gametes. And the gametes are the same for the male parent and the female parent. So here are the sperm, here are the eggs, and they're the same. Now if you go through this and you combine these gametes, these are the genotypes of the offspring. Now, what are the phenotypes of the offspring? Those are shown here. So there's 4 possible phenotypes. And these are the ratios of those 4 phenotypes. 9/16. So if you go through here and count these up and look at each square, you find that 9/16, or 9 of these squares out of the 16, are round yellow because they have at least one large R and one large Y. And the other allele is just putting a little line there to indicate it could be either big R or little r. It doesn't matter because as long as you have one large one, it doesn't matter what the other one is. So in terms of the phenotype, so round yellow seeds are going to be 9/16. The next phenotype is 3/16 wrinkled yellow. So if you go through here, you find 3 of these squares have 2 little r's and at least one large Y. So that's wrinkled yellow. And the next phenotype is 3/16 round green. So 3 of these squares are going to be at least one large R and 2 little Y's. So here's one of them, here's the other one, and the third one is right here. So round green. And then the last phenotype is 1/16 wrinkled green. And there's only one square that has that. 2 little r's, 2 little y's. So this phenotypic ratio of the dihybrid cross is 9:3 to 3:1. And that ratio told Mendel, that evidence indicated to him through the mathematics that he did that 2 genes are inherited independently of each other. Now let's work a couple of genetics problems. So the first one is to give the phenotypes for the following genotypes. So I've listed 4 genotypes here and asking for the phenotype that corresponds to the genotype. So the first one, we have 2 little y's. This is the gene, the Y gene for the seed color. So 2 little y's means that there's 2 recessive alleles and no dominant allele. So in the case of the Y gene, that's green. So this is going to be green seed. And as far as the seed shape, we've got a dominant capital R, and so that's round. So that first one is green round. Second one, we have a dominant large Y allele, and so we have yellow for the color. We have 2 dominant alleles for the shape, and so that's also round. Next one is 2 lowercase y's, so that's green. We have at least one large R, and so that's round. And then the last one is small y, that's green. 2 small y's is green. 2 small r's is wrinkled. That's the recessive trait for the seed shape. And the next question is about the fruit fly body color. We have big G being the gray or dominant allele. So if we have at least one large G, capital G, that's gray body, 2 small G's, black body. question is, what is the phenotypic ratio in offspring for the monohybrid cross? So we've got two heterozygous parents. What's going to be the ratio in the offspring from that? So what you want to do is draw a Punnett square. So you draw the possible sperm and the possible eggs. Big G, little g in both cases. You draw your Punnett square and you fill it in. So here we've got the big G and big G. Here big G from the egg, little g from the sperm. Here big G from the sperm, little g from the egg. And here we have two little g's from the sperm and the egg. So we can see from this that we've got three squares with at least one large G. So that's three gray and one black. So it's 3:1 gray to black is the phenotypic ratio. Next we cover the non-Mendelian inheritance. So in addition to Mendel's laws of inheritance, there are some additional phenomena. And so the Mendel's laws are not invalid here. They're just some additional things that we need to know about. So in the first, there's about 6 of these, and I'm going to cover 4 of them in this segment. The first is called incomplete dominance. So in incomplete dominance, the heterozygous genotype is intermediate between the two homozygous genotypes. So an example of that is the snapdragon flower color. So in snapdragons you have red flowers and white flowers. The red are homozygous big R and the white is homozygous little r. And in the heterozygous big R, little r, that combination is pink. So it's a blending of the red and the white. So you get that intermediate phenotype in the heterozygous condition. Another example of that is the LDL receptor. So the LDL receptor is a receptor that binds cholesterol, and normally in a healthy individual you have 2 capital H alleles, but some people have the heterozygous genotype, and so there's a big H and little h, and in that case they have intermediate level of the LDL receptor, so they have less of the receptor and therefore a buildup of cholesterol in their veins and some problems with that, some disease issues. And then there's other individuals that inherit 2 copies of the small h, and they have severe levels of cholesterol, and that can actually be lethal. And so that's just another example of the heterozygous condition being intermediate. So that's called incomplete dominance. And then the next one is called codominance. In codominance, you have 2 or more alleles, and the different alleles are both expressed independently of each other. So an example of that is the ABO blood groups. So in the ABO blood groups you have 3 alleles called A, B, and O. And in each person you have 2 alleles, and it can be either 2 A's, 2 B's, or it can be an A and a B, or it can be an A and an O, a B and an O, or it can even be two O's. So there's many different genotypes, but in each case, both of the alleles are expressed on the surface of red blood cells. So for example, in the case of the AB blood, you have both the A and B alleles being expressed, and so the A allele encodes produces a carbohydrate on the surface of the red blood cell called the A carbohydrate, and the B allele encodes the B carbohydrate. And in a person with AB blood who has the one A allele and one B allele, they have both of these carbohydrates on the surface of the red blood cells. So that's called codominance. The difference between codominance and incomplete dominance is that in codominance, the heterozygous or two different alleles gives you two different products, whereas in incomplete dominance, the intermediate phenotype from the heterozygous genotype is intermediate between the two homozygous genotypes. The third one is called pleiotropy. So pleiotropy is where a mutation in one gene has many effects. So you get many traits from a single gene. It's called pleiotropy. And an example of that is sickle cell disease. So in sickle cell disease, you have a single mutation in hemoglobin, in the gene that encodes hemoglobin. And this is the protein that carries oxygen in the blood. And one mutation, one amino acid change in that gene has many effects. So one effect is that the red blood cells kind of curl up or form a sickle shape, and that's one phenotype. Another is that the sickle shape of these cells causes the capillaries to clog, and that can cause damage to multiple organs. And that's another trait. And the third is that these sickle cells are attacked by the immune system, and so you get anemia or a lack of red blood cells because the sickled ones are being attacked. So many effects from a single mutation, a single gene. That's pleiotropy. The next one is called polygenic inheritance, and it's kind of the opposite of pleiotropy. It's where you have many genes affecting one trait. So an example of that would be human height. So height in a population follows a normal distribution or a bell-shaped curve. So most of us have about average height, but some people or a small number of people are unusually short, and a small number of people are unusually tall. And so you have this normal distribution, and that's because there are many genes involved, and so there's a continuum of or a spectrum of this trait, but it affects only one trait, in this case height. And here are a few other of the non-Mendelian inheritance. So first one is gene linkage. So gene linkage is where you have genes on the same chromosome and close together along the length. So let's look at an example. Here's chromosome 1. You have the paternal copy and the maternal copy, so these are homologous chromosomes. And we know that in meiotic prophase you get crossing over between the two homologous chromosomes. However, if two genes are very close together on the same chromosome, then the chance of a crossover right in between those is very small. And so these two genes close together tend to be inherited together. And whereas genes that are far apart, like AB, are far apart from C, in that case, there probably would be a crossover event between A, B, and C, but not a crossover between A and B because they're so close together on the same chromosome. In Mendel's case, he was looking at genes that were on different chromosomes. And his second law, the law of independent assortment, applies to genes that are on different chromosomes or are separated by a large distance on the same chromosome. But if they're very close together, that's called gene linkage, and they would not necessarily assort independently because of their linkage, their closeness. The next one is called X-linked inheritance, sometimes called sex-linked inheritance. And this is where a gene on the X chromosome becomes mutated. And an example of that is hemophilia. So this is a gene on the X chromosome that encodes a clotting factor. So this is a protein involved in clotting blood. And a mutation in that gene can cause that clotting factor to be defective, and so the clotting of the blood doesn't occur. Now normally this would be a rare mutation, and so most females would have only one X chromosome affected with this mutation, whereas the other copy of the X chromosome would be normal. So they'd have a healthy copy of that clotting factor gene, which would mask the mutated copy. And so they would be normal, they wouldn't have hemophilia, but they would be carriers of the mutation. Now in the male doesn't have two X chromosomes, he has an X and a Y chromosome. And so if a male inherits the mutated X chromosome from his mother and the Y from his father, of course, then he's got a single X chromosome with the mutation there. And so now there's no normal healthy copy of the X chromosome to mask the mutation on the X chromosome, and therefore the disease is expressed. And hemophilia is an example of that. And in the old times, before modern medicine, people actually bled to death from minor cuts because they simply didn't have the necessary clotting factors. Today we can supply the missing clotting factor because we can clone genes and express them and provide that. But this used to be a very serious condition. Another thing that is mentioned in chapter 8 is nondisjunction, and I don't think it's on the exam, but I think I should cover just for completeness. And that's the idea that occasionally in older egg cells, you may not have meiosis occurring properly, and occasionally some of the smaller chromosomes may not separate properly in meiosis. So you can have, for example, the chromosome 21 in an older egg might not, the two homologous copies of it may not segregate into the eggs, and both copies go to one egg and the other egg gets none. And so you have an egg with two copies of chromosome 21, and if that gets fertilized by a sperm with one copy of chromosome 21, now you have three copies of chromosome 21, and that's called trisomy 21, or better known as Down syndrome. So non-disjunction is a mistake during meiosis, and it's not really considered a non-Mendelian inheritance, but it does occur and is another type of genetic disease.